Welcome to Chapter 13, Biology students. It's all about genetics and biotechnology. In the 3 sections, you will see applied genetics, DNA technology, and the human genome. The big idea of this chapter is genetic technology improves human health and quality of life. There are 3 main ideas. The first one, selective breeding, is used to produce organisms with desired traits. Section 2's main idea is researchers use genetic engineering to manipulate DNA. And Section 3, genomes contain all of the information needed for an organism to grow and to survive. Here are your essential questions. And as you see, we have a review of hybrid DNA and codon, but you have almost 2 full columns of all brand new words, so start learning them now. Okay, Section 1, selective breeding. It's the process by which desired traits of certain plants and animals are selected and pass on their future generations. Desired traits can be passed on through hybridization and inbreeding. Hybridization involves crossing parent organisms with different forms of a trait to produce offsprings with specific traits. Hybrid organisms can be bred to be more disease resistant, to produce more offspring, or to grow faster. A disadvantage of hybridization is that it is time consuming as well as expensive. Inbreeding is the process of which 2 closely related organisms are bred to pass on a desirable trait to future generations. It can increase the chance of homozygous recessive offspring that may have harmful traits as well. So as you can see here, the purpose of hybridization is used for selective breeding, in particular animals and plants. So if you have a good swimmer and you also want them to have a good sense of smell, you would cross those 2 traits and hopefully you will get a dog that has both of those. There's some pros and cons also. Selective breeding is a pro, obviously. It creates new tastes in food. But some cons would be fungal infections in seeds and not able to be packaged as sold. And as you can see, inbreeding is a little bit different. So here we have a female puppy of a litter, and they are crossing her with the father of that litter. Another scenario that you might see is a female puppy of a litter being crossed with a male puppy, which would be a brother of that same litter to get the desired trait. And as you can see, there's a lot more that can go wrong with inbreeding versus when we use hybridization. So some pros of inbreeding: the result of an organism has desirable traits, which is what we're looking for. It's the whole reason why we do it. And it creates organisms of excellent quality. However, organisms can also end up having very, very strange mutations, and excessive breeding can lead to gene pool being limited, which could hurt your livestock. So here we have a test cross that involves breeding an organism that has the unknown genotype with one that is homozygous recessive for the desired trait. If the unknown parent genotype is homozygous dominant, all of the offspring will have that dominant phenotype. If the unknown parent genotype is heterozygous, the offspring will show a 1-to-1 phenotypic ratio. And that is your review for Section 1 of Chapter 13.